So my name is Allison Adams, and today I'm going to be presenting to you cancer immunotherapies, and specifically on the mechanism of T cell dysfunction and exhaustion. So the objectives for this presentation is going to be to understand what T cell exhaustion is and why it happens in cancer, to recognize the signs of T cell exhaustion, and then learn why T cell exhaustion and dysfunction matters, especially for cancer treatment immunotherapies. So we'll be going over the subtopic, the foundation information for understanding, what cancer is, what T cells are, and then going over metacognition, how I understand all of this, and then the conclusion. Sorry, I'm just going to take a drink of water. So for starters, what is cancer? Cancer is a disease known for its uncontrolled introduction of abnormal or unhealthy cells to our healthy body. Our body naturally is good at regulating these cells in our body and keeping them up in a healthy system, which is our immune system. Our innate immunity system is the first cellular defense against these cells, and should naturally kill them off, but sometimes this fails. It's crucial to understand that cancer is not a pathogen that enters the body, but it's one of our own cells that carry genetic mutation, which is going to alter the growth and the mechanisms and functions of these cells. If our body fails to target these cells and kill them off, it can lead to mass development of these abnormal cells that divide uncontrollably and can essentially destroy the body if they're cancerous. So the photo below is going to describe normal cells, which turn into abnormal cells, which we see by the yellow. It's yellow, or the orange epithelial. Those cells essentially multiply. What we see in the abnormal cells, that's just a benign. It's not invading any tissue yet. It's not invasive. And then we see it turn into malignant, which is when it's grown its own blood vessels, which is also known as angiogenesis. And then it's also going to invade the surrounding tissues as well. So we're going to talk a little bit more about malignant versus benign. So no, not all malignant cancers not all cell growths like this that are abnormal become cancerous or malignant. Malignant means cancerous cell growths that are uncontrolled. They have evaded surrounding tissue and can be spread through blood vessels and tissues. Benign tumors are cells that grow slowly. They don't invade the tissues and they don't spread, metastasize. They can become malignant, but it's very, very rare. So we see the photo on the left. We are shown benign versus malignant cancer cells. So the benign, which is non-cancerous, they're grouped clusters of neoplastic gland tubules, also known as tumor cells. They're encapsulated by this connective tissue, isolating it from surrounding tissue. That's going to be the top image. We can see that the cancerous cells do not mix with the normal gland tubules. They're not invasive, which in comparison to the malignant cancer cells, we see that the benign tumor cells still look very similar. They don't have as much mutations as the malignant ones. They look the same. They still grow faster than normal cells, but they still follow some normal cellular function. These benign cells don't carry as much genetic mutation as malignant cells, causing them to be less aggressive, invasive, and less harmful. So now we're going to talk a little bit more about the bottom image, which is the malignant cancer. Malignant tumors, which are cancerous. So malignant tumors show the opposite behaviors as benign ones. They are highly invasive, cancerous, and dangerous. In comparison to the benign tumors, we see that there's no fibrous connective tissue encapsulating these cancer cells. Nothing stopping it from spreading and intertwining with our healthier cells and tissues. we see that the shape and size of these cancer cells differ strongly from the benign ones. This is caused by de-differentiation, them losing their specialization. They don't have normal functions anymore and behave quite differently. These malignant cells gather more mutations over time as well as divide rapidly, causing them to appear different, grow uncontrollably, and cause them to lose their repair mechanism. So the different shapes aid them in invading different tissues, bloodstreams, or lymph vessels, which places they don't belong. I'm just going to take a drink of water. Alright, so on the left top image, these are going to be the common cancer sites in male and female. We see the lymph glands, the lungs, pancreas, bladder, prostate, rectum, bone marrow. And then on the female, the most common places are skin, breasts, kidney, colon, ovaries, and uterus. So on the middle image, we see top cancers in new cases, which are going to be 1.1 million new cases so far in the U.S. And then also we see the mortality rates of these as well on the further right, which is 9.16 million deaths. So almost half of the cases become deadly. So we see that lung cases are super deadly as far as how many cases they get, and then also colorectum. So there's a lot of breast cases, cancer cases as well, but not as many of them are deadly. So we're going to look at the key takeaways. Lung cancer is the most common and deadliest. Breast cancer is pretty common, but still has a lower mortality rate compared to lung, liver, and stomach cancers. Colorectal cancer, lung cancer, they're both common and very deadly. So now we're going to be going over the progression of cancer and basically the steps it takes in order to becoming very dangerous and spreading throughout the body. So the growth of cancer from normal epithelial cells to invasive cancerous cells. In order to understand this image better, it's key to remember that the red boxes are going to signify a genetic or molecular event, and then the yellow boxes are effects that the previous event had. So we see that these mutated cells all start off as normal epithelium, and then once the tumor suppressor gene is lost, they become excessive epithelial proliferation. So a tumor suppressor gene is a gene that aids in the regulation of cells from growing and dividing uncontrollably. But specifically the APC1 that is lost because there's different tumor suppressor genes. The adenomatous polyposis coli is going to control the growth and adhesion in the intestinal lining. And then the excessive epithelial proliferation is basically when there's a rapid multiplication or increase in these cells in order to produce more cells. This means that the oncogene RAS is activated, meaning that basically an oncogene is a gene that is mutated or an overactive version of one of our normal genes, which is also caused by uncontrolled cell division. That in turn makes it a small benign tumor, which it can stay that way. But if the tumor suppressor gene DCC is lost, which is the deleted colorectal carcinoma, it's going to aid in communication. So if that's lost, it's going to in turn become a large tumor. And then if the tumor suppressor gene P53 is lost, which is the one that's going to regulate DNA repair, detects damage, and then triggers apoptosis of the cell, which is cell death, killing them off if they're bad. If all of those signaling are lost, it's going to become very invasive, and rapid accumulation of mutations are going to occur, and then metastasis, which is basically when the cancer cells spread to other organs other than the origin. And then I got this information from the textbook, but it's also key to know that ChatGPT did help me understand these definitions better in order to word them better on here. So what causes cancer? Ultimately, cancer is caused by mutations, uncontrolled cell growth and replication in our genes. These genes are the ones that regulate cell growth, division, and cell death when it's needed. But what causes these mutations or increases the risk of getting these? We see genetic mutations, which basically are DNA that is damaged and doesn't die off, compromises oncogenes, tumor suppressor genes, and repair genes. We see that there are tons of environmental and lifestyle factors such as smoking, radiation, alcohol use, poor diet, low physical activity, and etc. These can all put you at an increased risk as well. Genetic inheritance is at 5 to 10% for the population of cancer. So 5 to 10% of cancer is hereditary, passed down from either parent. That puts you at increased risk. And then getting viruses or infections such as papillomavirus, hepatitis B, Epstein-Barr, and others have been linked to an increased risk of specific cancers. Aging is also another factor because your cells start to gather more mutations over time, which is why cancer is more common in older patients. So some of the key characteristics of cancer cells that it's important to know are that these cells stimulate their own growth. They ignore growth inhibiting signals, avoid death by apoptosis. They develop a blood supply, seeding from the site of origin to invade other tissues, constant replication to expand the tumor cell population, and then evasion and outrunning of the immune response. So some of the definitions just to go along with that basically are the growth inhibiting signals, which are going to prevent cell division when not needed or when damaged. Apoptosis is when programmed cell death for these unhealthy or unneeded cells. Angiogenesis is the building of new blood vessels in cancer in order for it to feed itself and spread. And then metastasis is when cancer cells have spread to other organs other than the origin. So since we are focusing on T cells and dysfunction, we're going to look at this image on the left, which is basically a melanoma, skin cancer, that's used a normal strand of GP100, which is a healthy protein. It's separated it, like as you can see in the image, we see the yellow, the red, and the blue. It's spliced that peptide and then put it together in places that it doesn't belong, which is the sorry, I'm thinking of the word. Mutation, sorry. Basically, this new protein only exists in unhealthy cells. So when the CD8 T cell recognizes a spliced GP100 peptide that's specific antigens CD8 T cell becomes activated, it kills the tumor cell. So the goal is for our immune system to spot all of these weird proteins that tumor cells have created in order to find tumor cells and destroy them. So not all cancer cells do need to be destroyed for it to be controlled by our immune system. I did a typo here, but basically, if our bodies can keep the tumor small, benign, or slow growing, it's considered control, not cancerous. I did not mean to put that word there. But the tumors that have been deemed successful are the ones that are cancerous, have evaded or manipulated our immune systems for survival and replication. So here is a image basically showing tumor-induced suppression of the anti-tumor T cell immune response. So how tumor T cells get past our immune system, they have tons of many tricks, super sneaky, and here's one way that they can and have. So in the image, we see different types of T cells, such as the CD8 T cell, the CD4 plus regulatory T cell, and then the CD4 T cell. So these all play vital roles in our immune system. First, the tumor cells, as we can see in the center, is going to release TGF-B, which is an immunosuppressive cytokine. The role of these cytokines are to weaken and stress out our immunity. The cytokines stop the CD8 T cell from killing the cells and CD4 T cells from activating the CD8 plus T cells. So next, the regulatory T cells release the TGF-B and the IL-10 cytokines because they're being pulled to help the tumors for evasion. The tumor cells, sorry. Lastly, as we can see, the CD8 T cells and the CD4 T cells are shut down. This is represented by the negative signs in the image. That means they're blocked pathways and can no longer enter that cell, and basically cuts them off. So it blocks these cells from spotting the tumor, which in turn is T cell dysfunction. So we're going to go over a little bit more of the dysfunction and exhaustion of these T cells. So after T cells fight a super long battle of keeping us safe from cancer, they're bound to get exhausted. When T cells are there but don't work, this is called T cell dysfunction. We can tell that T cells are dysfunctioning when they fail to kill cancer cells because of suppressive influences, which was shown on the previous slide. When they're worn out and tired, this is called T cell exhaustion. They've been overexposed to cancer cells for too long, and they start to see a reduction in cytokine production, cytotoxicity, and overexpression of inhibitory receptors. They also stop effectively killing off cancer cells and can no longer multiply. So what causes a T cell to fail in the first place? So we can see that T cells are tumors, or T cells can be in tumors. If they are, they tend to be suppressed and lacking of oxygen and room, so they struggle to kill these cancer cells. Immunotherapy's goal is to fix this and basically re-up their functioning. So T cells are most common to fail because of these reasons. They've either been around cancer antigens for too long, their fatigue causing them to be exhausted, or the brakes have been overused by the checkpoint proteins PD-1, TMI3, and CTLA4. This can lead to a lack of efficiency in immunotherapies, T cell exhaustion and suppression that is going to cut off the function of these T cells. If the tumor's microenvironment is suppressing the T cell, it's going to cause it to stop functioning properly. The functions stop due to nutrient deprivation and immunosuppressive cells like TREGs. The T cell exhaustion can lead to memory loss for the T cell, so it's going to stop recognizing these targets and stop attacking. It's also going to stop it from remembering these cells for the next time, these long-term memory cells. So immunotherapies take on this is that they want to awaken these cells. These killer functions, their ability to have these memory cells, block off exhaustion and expand and replicate these survival and functional T cells. So now we're going to talk a little bit more about cancer immunotherapy directly. So cancer immunotherapy is defined as treatments that boost or aid our immune system in the ability to fight off cancer cells. So instead of killing off these cancer cells directly with chemotherapy or other methods, we're going to talk about these three main types of cancer immunotherapy that aid in T cells overcoming exhaustion and dysfunction. So first, we're going to talk about immune checkpoint inhibitors, which is a drug that's going to take off the brakes of these T cells and stop them from overworking themselves. The checkpoint inhibitors block these switches, keeping the cancer cells from blocking it off. These are commonly used in cases of skin cancer, lung cancer, bladder, kidney, and head and neck cancer. Chimeric antigen receptor T cell therapy is a process where they modify the patient's immune system by removing these T cells, modifying them to express CAR, reinfusing them in order to fight off the cancer. This is going to help recognize specific cancer cells and have been used to fight off lymphoma or leukemia. And leukemia. And then lastly, one of the main ones is neoantigen vaccinations. This vaccination basically uses a mutation in the patient's tumor to create a vaccination for that person specifically. Cancer cells commonly carry mutations called the neoantigens. This vaccination is going to expose them or expose the patient to these specific neoantigens and help the T cells identify the tumor and attack it. Can you combine immunotherapies? Why would you do it? Is it recommended? We're going to go over that right now. So combining immunotherapies can actually be super essential for overriding the exhaustion and dysfunction of these T cells. Since cancer is super sneaky, it's going to find all ways to try and get around our immune system. It's safer to equip ourselves in every way we can in order for these immunotherapies to be effective. T cells cause a complexity of issues, the dysfunction and exhaustion of these T cells cause tons of issues, not just one. So in order to effectively reverse and target all of these, we're going to have to use multiple methods. So what these combinations, two or more can do is basically it's going to attack the different layers of suppression all at once. It can waken up these T cells, fight off exhaustion, activate these T cells, and charge the old ones simultaneously. And then also it's going to prevent the cancer cells from evading before getting targeted by these newly charged T cells. Some example combinations and their accomplishments are when you add 2 checkpoint inhibitors together, it's going to awaken the max amount of T cells to block off 2 different switches. If you use a checkpoint inhibitor immunotherapy and then the TGF-beta or the IL-10 blocker, since TGF-beta and IL-10 are suppressive signals, blocking them helps T cells stay alert and functioning. And then if you do the checkpoint inhibitor and the neoantigen vaccination, it's going to, basically the vaccine is going to train these T cells while the checkpoint inhibitor aids them in staying active inside of the tumor. So I did pull from 2 journal articles that I found, took their key, not differences, just their key points, and I kind of wrote them down just to explain this topic a little better and what their specific take was. So the first article that I read on was T cell dysfunction and exhaustion in cancer. So this article's key focus was to explain the biological and molecular reasons as to why T cells become exhausted and dysfunctional in cancerous environments. The most important components were that T cells in cancer in this article are defined as a state of sustained inhibitory receptor expression and transcriptional reprogramming, which reduces their ability to kill and promote the progression of the growing tumor. The dysfunction factors caused by intrinsic factors such as TOX, NR4A1, EOMs, and metabolic changes. Epigenetic changes reinforce the exhaustion by altering DNA accessibility and the gene expression in these T cells. They also state that the microenvironment of the tumor can add intrinsic pressure and shut down T cells from functioning. The checkpoint inhibitors can reignite the T cells that are exhausted. The best results are from combination therapies that target metabolism, epigenetic, and neoantigen vaccinations that we previously talked about. So the ending of the article is going to conclude that there's still many challenges with immunotherapy that need to be studied. It does state that a lot of these immunotherapies have been focused around T cell exhaustion and dysfunction, so that we should see some breakthroughs in the next few years. They're hoping with further studies of novel targeted therapeutics that it should improve the efficiency altogether of these immunotherapies. It concludes stating that further studies should focus on turning cold tumors to hot tumors by aiding T cells in infiltration. The second journal article that I came up on was Reversible T cell dysfunction and exhaustion in cancer. This article starts off by focusing on why T cells become dysfunctional, stating that it's due to chronic antigen exposure that causes less cytokines to be produced, the reduction of proliferation, which essentially the reproduction, and the lack of ability to regulate the inhibitor proliferation, which essentially the reproduction, and the lack of ability to regulate the inhibitor receptors, which are the PD1, CTLA4, TIM3, LAG3, and TGIAT. They talk about the ability that these cells have in order to regain their function after exhaustion through these immunotherapies, basically reversing them. Noted that suppression of the immunity against tumors are caused by these regulatory T cells such as TREGs, MDSCs, and TAMs. They point out that because immune checkpoints are often not fulfilling, combination therapies such as PD1, IL-10 can lead to a stronger restoration of these T cell functions. They close out by stating that T cell dysfunction and exhaustion caused by tumors can be reversed through combination immunotherapies. It'll counteract the mechanism of the tumor. He also states that he believes personalized immunotherapy specialized to each patient is more effective, but it poses the challenge of finding the specific immunoregulatory and oncogenetic pathways that drive each patient's T cell into exhaustion in the first place. So to wrap all of this up, metacognition, how I've kind of been processing all of this, basically everything that was in this presentation has kind of been a basis, the foundation of this course in general, because it does have to do with our immune system. So I think it's best to talk about my metacognition over the timeline of this course. This course has been so packed with information in such a small timeline. I'm definitely not used to this. This is my first time taking a summer course. Oops, sorry. So it's definitely a challenge for me to get used to this, but all in all, I mean, I found it hard to wrap my head around certain concepts like TLRs and then the different types of cells in the body, their functions, kind of the basics of what we learned in the beginning. And then over time, it got a lot easier. When I started note-taking and stuff, I feel like I definitely comprehended it more. The slides alongside the discussions definitely helped me a lot with comprehension as well. So in conclusion, now that we've gone over the basics of cancer, how it develops, the toll on its body, etc., we've set a foundation for understanding the T cell's exhaustion and dysfunction. We've also learned how cancer immunotherapies can combat this in order to reawaken these cells to function through different therapies. And then in conclusion, there are so many new therapies that are being studied on top of the ones we've already covered. They're working on making a breakthrough in our healthcare and cancer. They're currently doing many clinical trials and studies regarding metabolic reprogramming and furthering studies that we've covered. So these are the resources I used. All of the images, the links are right here. Tons of them were from the textbook. And then for the scientific information, I did get a lot of it from the textbook and our slides for the chapters. I also gathered information from the 2 journal articles. And then I did use ChatGPT to revise a few of my slides and to help me kind of break down the information and understand it better and basically aid in how I was able to comprehend and present this. Thank you for watching.